In this video, we are going to explain the endosymbiotic theory, and how it relates to the horizontal gene transfer. What the endosymbiotic theory is about. This theory describes how a large host cell and ingested bacteria could easily become dependent on one another for survival, resulting in a permanent relationship. Over millions of years of evolution, mitochondria and chloroplasts have become more specialized, and today they cannot live outside the cell. And now, what does endosymbiosis mean? Endo means within and symbiosis means together, so endosymbiosis means an organism living in a symbiotic association with another, specifically by living inside a host cell. One of the great evidences of this theory is that the mitochondria and the chloroplasts have their own DNA, which is transferred to the cellular nucleus, but how it happens? Horizontal gene transfer is the answer. Exist three ways to make this transference. Transformation occurs when naked DNA is released on lysis of an organism and is taken up by another organism. Another way is by transduction. Antibiotic resistance genes are transferred from one bacterium to another by means of bacteriophages. And finally, by conjugation, occurs by direct contact between two bacteria. Plasmids form a mating bridge across the bacteria and DNA is exchanged to become an active nuclear gene and return to the organelle as a functional protein. An organelle or gene must first be assimilated into the nuclear genome. But how? Through comparative and experimental, nucleic acids can be shuttled out of mitochondria through membrane transporters. Either RNA or DNA can serve as transfer intermediates. Experimental studies also suggest that mitochondrial DNA can find its way to the nucleus independent of an RNA intermediate. Now, why those bacteria of free life keep in the cell and never separated? The bacteria received protection and food while they were able to provide molecules of ATP and molecules with reducing power obtained by their phosphorylation system, which after successive divisions, gave rise to the eukaryotic cell. In this way, both the cell and the bacteria would benefit from its new symbiotic relationship. If chloroplasts and mitochondria come from bacteria, what types of bacteria would they be? Much evidence supports the mitochondrial genome originated from within the eubacterial specific call. The proteobacteria are the closest identified relatives of mitochondria. Endosymbiotic theory posits the origin of the chloroplasts. A cell ate a photosynthetic cyanobacterium and failed to digest it. There is a great amount of evidence that verifies this theory, making it nowadays scientifically accepted and almost taken for granted. Among the main evidences we have: first, they are surrounded by a double membrane, which agrees with the idea of phagocytosis. Second, mitochondria and chloroplasts contain covalently closed circular double-stranded DNA, just like prokaryotes. And third, in mitochondria and chloroplasts we find ribosomes 70s, characteristic of prokaryotes, while in the rest of the eukaryotic cell, the ribosomes are 80s. Naturally, we are accustomed to hearing about the use of nuclear DNA for different studies. However, on many occasions, especially in forensic contexts, the samples have been exposed to such adverse conditions that the study of nuclear DNA is not possible, because it is very degraded, or in small quantity. In these cases, the mitochondrial DNA can be used since the probabilities of success in the analysis are much higher than those of the nuclear marker analysis. Two of the main advantages of using mitochondrial DNA over nuclear DNA are: first, mitochondrial DNA is present in a very high number of copies per cell. And second, the uniparental inheritance and the absence of mitochondrial DNA recombination allow the comparison of members of a lineage that share a common ancestor through maternal root, providing the basis for the identification of biological evidences. Thanks, hope you enjoyed.